presentation of the season. Miguel is here for his 6-year promotion and tenure review, and he'll be talking to us about ecotoxicology in birds. So make sure I got it.
Thank you, Gary. Well, I'm going to start with basically acknowledging the assistance of all my students because most of the stuff I'm going to tell you is basically part of the work they've done over the last 6 years. Saria worked with me on a project in Chapala, contaminants in Chapala Lake, and then she was instrumental in a stable isotope study in Colorado County here in Texas. Chelsea, currently here, she's finishing up the study with the Aplomado falcon in South Texas. Hannah was very good in helping me in a project on insecticides, neonicotinoid insecticides and the impacts on bowhide. I'm going to touch on one of those projects in more detail. And then Alejandra sort of follow up what I've been interested in for the last 30 years, addressing contaminant issues on migratory birds. And so she did a study with neotropical migrants and Texas, Mexico, and Costa Rica. I'll touch a little bit on that. Chris got a master's degree working with neotropic cormorants in the Trinity River watershed. And then Jules and we worked on an interesting project regarding, we were trying to determine whether we could use stable isotope techniques to diagnose the contribution of E. coli from water birds to watersheds. We're still working on that. I think we have a good track. We have a good indicator, and I'll talk a little bit about that. So the approach I'm going to follow is very similar. I guess I'm supposed to talk about research, teaching, and service. I'll basically talk about the research. I'll have one slide on teaching, a couple of slides on service, and that'll be it. I should give a little brief overview, mostly because most of you don't work in this field, and so that you get a little bit familiar with what is it that I do. Wildlife ecotoxicology, basically we use birds as indicator species to understand or address the effects of contaminants on birds. I think birds are good indicator species because they are at the top of the food web, so they could bioaccumulate, biomagnify contaminants. We have a lot of birds in aquatic environments where we usually find those contaminants, so that's where I focus for the most part. Part of the focus is also on persistent organic pollutants, mostly because, well, one thing is because they last longer, so you could track them. They don't disappear as fast. They disperse long distances. And because they are lipophilic or hydrophobic, they accumulate through the food web, so you could easily basically use indicator species and then address issues regarding a community or a particular ecosystem. Lately, in the last 20 years or so, there's been a lot of noise on endocrine disruption, mostly because there have been issues of feminization, masculinization, effects on reproductive success. So we focus on some of those contaminants that are endocrine disruptors. And this is just a slide showing how the contaminants could easily go up the food web. But the main point is to highlight that you start with a very small concentration. Like in this case, you start with a very, very small concentration, and then by the time you get up at the top of the food web, if you divide those values, the concentration has been gone up thousands of times. And that is why it will have a stronger effect on the top predator. So we are dealing with contaminants that have been banned, at least in this country, for 30 years or more, 40 years. Since the 70s, like polychlorinated biphenyls. No longer in use, but there's so many out there in the environment, they're still causing problems. These were used in electrical transformers. Still a lot of them are in dumps, but they are highly persistent, highly toxic. They have created lots of different problems in wildlife. So they are part of what we call legacy pollutants. And this slide from Science in recent years shows that they remain a major threat to marine mammals. And actually, marine mammals are probably the most contaminated vertebrates in the world. All of them because they are long-lived. They basically apex predators. They accumulate lots of contaminants. More recent contaminants and very close to home, the Brighton Fire Training Facility. I've been trying to study issues around that plant for a long time. Unfortunately, nobody wants to fund me those studies. They release a lot of chemicals that are highly toxic, fire retardants. They use them to extinguish some of those fires. But we also use them in plastic, wood, paper, in clothes, whatever we need to protect for fire, computers. And a lot of them have actually gone into the list of contaminants that should be phased out and have been phased out. More recently, we use chemicals that have this kind of weird structure, lots of carbons and lots of fluors. That's why they're called perfluorocarbon structures. But they are used everywhere, carpets, and at home, and surfactants, and upholstery, and fabric, and packaging. And they are also similar to the others. They persist for a long time. They are highly toxic, and some of them are being placed in the list of originally called the dirty dozen. I think this body is close to 25 now, compounds to be nominated to be phased out for no use in the whole world. And why do I bring this up? Because although they are legacy contaminants, they're still a problem. We still see issues. For example, this paper on songbirds in Michigan about 4 years ago, they found that the highest DDT concentrations ever reported in songbirds were detected in Michigan. So some of us are looking for them in Central America, but they are close to home. Then the condor is an endangered species that we've been trying to recover for a long time. We're still finding that they're having problems because of DDT still present in California and Arizona. And more recently, even you will see articles where they find bats that have been poisoned in Montana, where you wouldn't think we have any of those contaminants. So the issues are still around, they haven't gone, and because of the long persistence, these contaminants are there. So much for that interview or overview. In the last 6 years, some of the main focus of my research is the contaminant monitoring studies. So we have published a few papers with some of my students. Sandoval et al., we just published a paper on neotropic cormorants. Hanna, that's her et al., published a couple of papers on neonicotinoids. And Eric Schmud, actually, this student, distance education student, I was able to manage him so he could write a paper on metals in bowites. That was interesting. And then Alejandra on neotropical songbirds. And I did a review a few years ago on the current status of persistent organic pollutants in North America that appear in environmental toxicology and chemistry. Briefly, a couple of slides of Alejandra's project. She basically was interested in looking at accumulation of pesticides on songbirds while they migrate to Central and South America, although we didn't go to South America. So she basically focused a study in 3 locations, several locations in Texas, and then in the Yucatan, 2 areas in the Yucatan, and then in Costa Rica. And over a period of 2 years, she collected samples, and the most interesting thing that I want to show is what we have found 20 years earlier, or 25 years earlier, that we have more problems at home than sometimes in the South in terms of contaminants, because we use a lot more pesticides than they use in other countries. You could have some DDT, it's an isomer of DDT. You have some DDT values here in verse collected in Texas in the fall, Yucatan in the winter, Costa Rica in the winter, and Texas in the spring. The only thing to highlight here is the sort of reddish bars. So you can notice that, first of all, to point out that good news, contaminant concentrations are very low in these birds. They're small birds, as it should be expected. But secondly, the most important thing to highlight here is that if you look at Texas, the resident species in the fall and in the spring show the highest residues relative to Yucatan and Costa Rica. So in essence, just basically telling us that it is true that there are a lot of insecticides used in South America. There have been isolated incidents, but in terms of these contaminants that were used in the past, probably the problems are closer to the south, in the border between Texas and Mexico. I have been working with the Perkin Fund for the last 20 years, trying to evaluate the potential impacts that contaminants might have on the recovery of the apple falcon in South Texas. And for the last 20 years, we have been collecting eggs that do not hatch, adult eggs, and I just basically saved them for a good time until I got Chelsea, convinced her to work and do the analytical chemistry of those compounds. So we have analyzed the eggs that did not hatch for a period of almost 20 years, 2001 to 2018. And it's very interesting and excited that this beautiful bird, it appears to be on its way to recovery in South Texas. My guess that is that over the few years, unless the neonicotinoids hit them, they might be doing well. Just a little thing to notice is that there is a graph here that shows concentrations of DDE. This will be the contaminant, and this will be the thickness of the eggshell, and that is a good indicator of accumulation of contaminants. You will notice that as the concentration of DDE increases, the eggshell thickness tends to decrease. Now, this is not a very significant decline, but it does show a tendency to show decreased thickness with increased concentrations of DDE. Now, this is a phenomenon that we have seen for a long time, and it's occurring, so it goes to show that that compound is associated with eggshell thinning, but in this case, the effects are minor. The most interesting thing also to notice is that we look at the thickness of the eggshell over time is showing a slight increase, indicating also again that in terms of contaminant impacts, these birds do not seem to be affected currently by organochlorines or other persistent organic pollutants. And the populations are doing more or less okay. The Perkin Fund has been monitoring the populations since they started releasing birds in 1995. And you can see that since 2000, more or less, the population has been sort of stable around 80, except for this dip in 2018. What happens is Hurricane Harvey in 2016 hit them hard. So it killed some of the birds from 2017. So by the time they do the survey in 2018, the population was down. I was told, however, that the few that survived the Hurricane Harvey did very well in productivity, so our numbers should be up next year. So that's where the population is going. All right, so that's contaminant monitoring studies. Another chapter in the last 6 years has been my collaboration with colleagues in Mexico. We have been able to train some students and publish some papers. 4 of these papers are from students that we train. One of them is Saria. I've been working in Chapala Lake, which is the largest lake in Mexico. It's a shallow lake, highly alkaline, and receives a lot of contaminants from the Lerma River, which is probably the only tributary of Chapala Lake. And one of the main reasons I've been working in Chapala is because I'm interested in contaminant issues in Chapala, but also the group that I work with is located here in Jiquilpan, and that is my hometown. So it sort of works for me to go there to work with them. So I'm not going to talk about any of those studies except for one thing that was interesting. One of our articles was highlighted in the front page and the cover of Environmental Science and Technology, a journal published by the American Chemical Society. Chapala, by the way, is one of the primary winter grounds of white pelicans, and that is supposed to be a Ramsar site, and it should be protected, but we know very little about the pollution in this lake. Okay, now I'm going to talk a little bit more detail about the study that we did on neonicotinoid insecticides and the potential association with northern bowhhites in Texas. This study was triggered because there's been concern that the bowhhites have been declining in Texas for a long time. And the state has some money, the quail decline initiative, and we were able to dig in and get some money to do these studies. And I propose working on neonicotinoids because currently they are the highest used pesticides in the world. They are supposed to be specific for insects because they affect the receptor that is specific of insects, but studies have shown that they also affect birds in many different ways. In Texas, the most used neonicotinoid is imidacloprid, and by the way, that is in all over the world. And except for these two, thiacloprid and 9-10-pyram, the others are still used. By the way, these compounds are no longer used in the European Union, and I'll tell you why later on. But we still use them, and we highly use them in Texas. So the mode of action of these compounds is through the nicotinic acetylcholine receptor in the insects. So affects the transmission of nerve impulses, so it leads to tremors, paralysis, and death. But studies show that they also affect reproduction, development, behavior, immune suppression of birds, and they have indirect effects. There are studies, recent studies, for example, this was done in Japan, that they have shown that this one clothianidin affects the reproductive function of quail and leads to oxidative stress. So there's a lot of evidence out there that suggests that there could be a link, a connection to the use of these compounds with declines not only of bovines, but a lot of grassland birds. Texas began using these or applying these neonicotinoids in the mid-90s. And you may be familiar with this, all this color map shows where these neonics have been used. But the county where most of the neonicotinoids, or the highest use of neonicotinoids has been, is this in South Texas, which is Hidalgo County. So what we did is collected a lot of information on pesticide use, quail abundance, and then other variables, and we correlated those data over time. And I'm going to tell you what we found. First of all, collection of data indicated there were areas of high use, moderate use, and low use in Texas, as you can see here in the map. I'm going to try this because I'm going there too much. Okay, so it's it doesn't work there. So you have then low, high, and moderate use, but the only thing that I want to notice is, so here is where neonicotinoids began to be used. Notice that the first years, the use was sort of minimal. But notice how around 2003 or 2004, there is a spike. And all this data have been collected, by the way, by the U.S. Geological Survey. The interesting thing is that the sharper decline in bovine abundance in Texas and many areas in the south began around this time, around the mid-90s. Could be just a coincidence, but that's also something that we were looking at. So we collected information from two sources, the Breeding Bird Survey and the Texas Parks and Wildlife Department. And this information is collected differently. The Breeding Bird Survey is coordinated by the U.S. Geological Survey, and all these little things you see in the map, all those are transects, like this one you see in the study areas, that are about, in average, about 41 kilometers, okay? And what we did is collect the information from each one of these sites and expanded it to half a mile each way, no, half a kilometer each way. So the segment was 1 kilometer wide and 41 kilometers long. And for the Texas Parks and Wildlife Department, the average transect was 32 kilometers and then 1 kilometer wide. Now, the Breeding Bird Survey is conducted in a way that they select that transect, then they select 50 different points, and the people that are doing the survey stop at each point and then count the birds they see for 3 minutes. So 50 different stops, 3 minutes a count. The Texas Parks and Wildlife, the way they do it, they drive a vehicle at 20 kilometers or 20 miles per hour, and then they count whatever they see. So obviously the information is going to be significantly different, but those are the two databases that are used to correlate abundance in the United States and in the States. So we use the information from those two places and then try to link it to pesticide use. So we collected bovine abundance and then linked that with summer drought index, breeding season precipitation, summer mean maximum temperature, total cultivated cropland, and total developed area. Now, we did that for each one of these segments, the segments that you do here. So we extrapolated, we calculated the amount of agriculture, the amount of developed land that was in there, and then the amount of potential neonicotinoid use in those areas, and then correlate that with abundance, okay? So, in addition to that, we selected three sites, 3 locations, one in the rolling plains, one in the Gulf Coast prairies and marshes, and one in South Texas to collect some quail that we later on analyzed for neonicotinoid residues. So, because the neonicotinoids began to be used in the mid-90s, and we had quail abundance data in 1978, we decided to split the analysis into 3 things. Before neonicotinoid use, that would be this green area, then post, that when neonicotinoid began to be used. But because there were 2 kind of periods where neonics were being used in Texas, we selected this period as a period of basically low use, and then this period from 2004 to 2012, a period of lay use. So we have 3 segments of data to analyze to look at correlations or relationships between quail abundance, neonicotinoids use, and all the other variables. So this may look like a mess to you, but I could explain it to you later on. I don't think I have time here. But we have basically 7 ecoregions in Texas, from Cross Timbers to Edwards Plateau, Gulf Coast prairies and marshes, High Plains, Piney Woods, Rolling Plains, and South Texas. And we have 2 data sets, BBS, Breeding Bird Survey, and Texas Parks and Wildlife. So you have that. And then we had 3 periods, pre, post, and late. We had to hire or ask a statistician to join us to do this analysis. So a good friend of mine who's a well-known statistician agreed to cooperate with us, look at the data, and help us to figure out what would be the best models that we could feed to analyze the data. So the quail abundance data have a lot of zeros. And because have a lot of zeros, they are zero inflated. So that was a particular problem. So we had to deal with zero inflated data. And the proposal was that we had to first fit the model using general linear models to all those variables, select the ones that were the most parsimonious through stepwise regressions, forward and backward, and then once we selected those variables, fit them to models that were able to deal with zero inflated values. And those models are the additive model, the zero inflated model, the Hurdle model, and the general linear mixed model. After we rattled that, we selected those that provided the best AIC criteria. If you follow me, then that was able to give us a lot of information for each one of those ecoregions. I'm just giving you here an example of 2 other ecoregions. I could send you a copy of the paper if you want to see them, the complete ones. But the only point I want to make here is that we have all the variables that we look at, and here I'm going to have to put my glasses because I can't see. So we have summer drought index, breeding season precipitation, summer mean maximum temperature, total cultivated cropland, and here is the column that we are interested in the most. This is the total neonicotinoid use. We found out that a lot of the models, most of the models were negatively and significantly associated with bovine abundance. In other In other words, neonicotinoids were the ones that explain the decline in numbers of abundance the most. And I'll show you that compilation. Of all those variables, you look at this table, you have those that have a positive association. And as expected, the summer drought index had the highest positive association with bovine abundance. But in a negative way, the total neonicotinoid use had the highest negative association. It was 62%, okay? So when you look at the bovine abundance data just on its own, on those 3 periods that we determined, pre-pest, post-pest, and late pest, notice that the bovine was already declining, but not as much in the 70s. If you look at the second period, the decline seems to be a little bit more significant, but after 2000, if you look at this period, it looks like the decline is more significant. And that is the period where the neonicotinoids began to be used at greatest amounts in Texas, okay? So we conclude from that study, and the other we had, is that neonicotinoids may be contributing to quail decline in moderate and high-use areas. Here's all the state. And this is also corroborated by residues that were found in some of the samples that are undamaged in tissues that is significant, that is consistent with poisoning or neonicotinoids. So after this, we propose to conduct a thorough ecological risk assessment in the state to really come up with a model that would indicate whether bovites in reality were linked to the use of neonicotinoids. If that decline was linked to the use of neonicotinoids in the state, but it was not funded. It was probably research that wouldn't be fun to find in Texas because we use a lot of those compounds in agriculture. Well, that's just my take anyway. So, all right. So if you're still with me, we're going to move to another study that is probably more ecological, estimating diet sources of the endangered species, the Alwater prairie chicken, using stabilized stops and Bayesian mixing models. The Alwater prairie chicken is an endangered species that is currently mostly present in 2 areas, which is in Colorado County and in the preserve close to Galveston. Very slow numbers. But you could see that basically since the 1930s, the populations were basically totally declining. And now the population is basically maintained through a breeding program. So the Fish and Wildlife Service was interested in determining what's happening because the chicks, they have a high mortality. The chicks die right after 2-3 weeks, and apparently they haven't been able to determine what is the real cause. A lot of hypotheses, but one of the most recent ones is that the insect abundance in the area has decreased so that the birds do not have enough food available to survive. They will look at other issues like predation, all sorts of different issues, but we got involved in the diet study using stable isotopes, and that's what I'm going to show you today. The reasons they think that the species decline is like what you hear from most of the other studies. There have been a lot of changes in the grassland. Fire ants, there's always a culprit. There's actually the books, biology books show a nice association of bovine decline with the fire ants. So it's possible that they had a strong impact in Alwater prairie chickens. But of course, nobody could deny that agriculture is always a problem because of the use of pesticides. But the recovery plan from the U.S. Fish and Wildlife Service says that the single most important factor that limits progress is the poor survival. So what is it that we did? We collected all sorts of different potential food sources in the Colorado County. And we follow the advice of the biologists there. And we then focus on some of the foods that they thought the unwanted prairie chickens were eating. So we're trying to sample good sources that could help our model. So lots of forks and grasses, lots of arthropods, and then from the consumers themselves, we collected blood, feathers, and feces. But we also wanted to see whether there was a difference, historical difference, between prairie chickens that existed in the wild, like in the 30s or before that, and the current prairie chickens. So we collected feathers from museum specimens and feathers from current chickens in the wild. We use stable isotopes because they have been used a lot in ecological studies because they are indicators, particularly carbon and nitrogen, indicators of diet. And we use the ratio of a heavy to a light isotope because that's what is used. And the only difference between, say, 13C and 12C is 1 neutron, so one is heavier than the other. But the ratio is recognized as a delta, so we will see delta 13C, delta 15N, so it will be the ratio of the heavier to the light isotope. The theory is that as the food moves from one source to another, there is discrimination of these isotopes, either an increase or a decrease, depending on assimilation and elimination rates. So we measure carbon and nitrogen in all the, I don't know, over 300 samples that we have. And this, as you can see, is a mess. And this is basically just the map of everything, delta 13C, delta 15N. C3 represents C3 plants, C4 plants. I will be the insects, and then the S will be the spiders. We separate the spiders from the insects because they tended to have higher nitrogen values. But according to the literature, if we wanted to use the mixture model with this data, it would be difficult because the literature says that when you have sources that are not separated in space, it's a lot more difficult to determine what is going to be the diet of that particular animal or organism. So based on this, what I decided was to use a program, or not a program, but a function available in Jump and SAS that allows you to organize your data in clusters using a K2 clustering procedure, where it selects some centroids based on the distribution of all the data. And in this case, you will see 4 clusters. And then through an iteration procedure, locates all the values that are closer to that cluster and then puts them together. So what this does, it facilitates the distribution of these potential sources and actually helps you to really determine what is going to be the diet more specific, because in this case, we are looking at partition or discrimination of carbon and nitrogen, not exactly if it's an insect or in a plant. In the end, we want to know if it's an insect in a plant, but for the model just wants to know what is the closest source based on the carbon or the nitrogen value. So we have 4 clusters, and those were going to be our sources. And then I categorize those clusters. So what you could see that, for example, cluster 1 is very rich in nitrogen, the highest levels of nitrogen. And then you see cluster 2 is very rich in carbon. And then there is a distribution of others. But you can see that the cluster 1 has a higher percent of spiders, some insects, some C3 plants, no C4. We notice that cluster 2 basically has also some spiders, lots of insects, and all the C4 plants that we have. So using these as sources, I could then determine what would be happening to how can we tell the diet, how can we tell it apart, okay? So we use then a mixture model that basically uses the biotracer data to estimate the proportions of prey that contribute to the mixture. This mixture model also uses Markov chain Monte Carlo iterations to basically come up with probability distribution, the density function of the mean value of the particular source that you're going to determine. So this model could run a small chain, a long chain, or a very long chain that could last for days or for hours. A normal chain will take 100,000 iterations, and it will give you, will go through the chain that it goes to give you the most conversions when it's telling you that the distributions are well determined. So, believing that this model has been validated, has been used by many people, I think the main thing I have to be sure is that I have the right set of data to run the model. So to get the right set of data, I need to also get what we call discrimination factor. And this is one of the main problems to running these models, because sometimes we don't have these discrimination factors and we take them from the literature. In this case, knowing that we had captive prairie chickens in zoos, I contacted the Houston Zoo, and we determined, or we obtained samples from them of a lot of feathers and the food that they were given in the diet. So from chicks that were growing in captivity, or samples that were growing in captivity, we analyzed the diet, we analyzed their blood, and we analyzed their feathers. And then calculated the discrimination factor, which basically is the difference in delta 13C or delta 15M values between the consumer and the resource. In this case, will be the tissue and the diet. So the blood and the diet, the feather and the diet. So the model is going to need these discrimination factors to go to a process of determining the most likely sources based on the consumer values. So we also need to have all the consumer values, and this is just an isospace plot that show you all these little dots that are values, the delta 13C and delta 15M values of blood samples. So these are samples collected from the prairie chickens. This plot is important because for the model to run effectively, you need to make sure that your consumer values are inside a polygon. A polygon indicated that the model is actually really capturing the information from all the potential sources. And in this case, I have cluster 1, cluster 2, cluster 4, and cluster 3 is more into there because we had a very wide distribution of insect values, delta 13C, delta 15M values. But at least this isospace plot was telling me that this data set is effective and is adequate to be run with the mixture model. So running the model, then I get some results. I just show you that, you can't read that, but the model gives you a posterior density plot. This is, for example, for museum feathers, that is telling you the proportion of the diet. For example, in this case, it's saying that cluster 1 and cluster 2 are the ones that contribute the most to the diet that is assimilated in feathers of these prairie chickens. Okay? So I could calculate the median for that value and then calculate the proportion of insects or plants that are present in that cluster. So doing all those calculations, I get with a predicted diet, a calculated percent for the prairie chickens based on the mixture models. What is it that I got? I'm going to focus on the summer and fall values because I have some of my questions on the winter values. But what the model then tells me is that the diet of these, based on blood, the diet of these chickens in the summer is 60% insects, close to 10% spiders, 28, 22% plants, and very little, a little bit of C4 plants. Now, the winter is a little questionable for one thing. We couldn't collect insects in the months of December or January. We tried, maybe our effort was not good enough, we just couldn't collect them. So the argument is that the insects are not as available in the winter. So if the source is not present, well, the birds are going to eat something else. So in that sense, the model is just basically using a lot of the sources to say, well, if insects were available, they will be eaten this much. But they are not. Now, I'm going to show you other data that are still a little contradicting with that. But anyway, so The year-round is just a year-round estimate, but the summer and fall, to me, are the most important data, corroborating that they feed a lot of insects, a little bit on spiders, a little bit on see-through plants. Now, the data from feathers are a little different, and they indicate that the assimilation of carbon and nitrogen in the feathers is not similar, as some people had indicated, and that it's a little bit different because you can see that based on feathers, you are predicting that they probably feed a little more of insects. But the interesting thing, and I'm going to make a comment later on, is the slight difference in the percentage of spiders between contemporary, that is the recent feathers, and museum feathers. Now, there are some papers out there in the literature that argue that the insect densities have gone down over time because of the use of pesticides in agriculture. And also there are studies that suggest that the spiders have decreased in many areas. And this would indicate that that is the case. It will make sense why now the current prairie chickens in the wild are feeding less on spiders than in the past, but more information later. So when I transfer or translate the insects to the different types of insects that these animals ate on, the predominant insects they fed on was in Orthoptera and Hemiptera, a little bit on Coleoptera. And this is very interesting because these were the most common insects that we sample. So this information is coming from the model. And what we sampled were most Orthoptera and Hemiptera and Coleoptera. Okay? And analysis of fecal material indicates that the most common insects that we found were Orthoptera, Hemiptera, and Coleoptera. And as I indicated, also our efforts collecting gave us the most Orthoptera, Hemiptera, and Coleoptera. So we had a model telling us that, our collection efforts telling us that, and the fecal analysis telling us that. Now, one interesting thing is that these feces were collected in January. So that would be the winter. So if we couldn't find the insects, why is it that the fecal material is given as this high content of insects? So that is something that I still need to analyze what's happening. Okay? So, what is the discrepancy? Well, the only studies that are on diets of the average prairie chickens were done on fecal material, on feces. And one was done by Kogar. I guess I was a student of Dr. Silby in the 80s, Lehman in 1941, Kessler in 1978. All of them indicate that the prairie chickens feed primarily on foliage, on vegetation. Like Lehman says, mostly carnivorous, higher use of insects in summer, plants, and insects annually. Kessler is the one that says in summer about 71%. I think probably Lehman and Kessler are a lot closer to our data for the fall and the summer. We still have to resolve the difference in the winter. Now, we are looking at values that have been assimilated in the animal. So what the animal got in the blood and in the feathers from the food. They are looking at the leftovers from the fecal material, what it was not assimilated. So we have a difference there too that we need to explore. Okay? So, anyway, I think I'm running out of time and I don't want to bother you. We've also been doing several studies that I indicated to determine to what extent we could use to find a stable isotope technique of fecal sterols to determine the contribution of E. coli from waterbirds to watersheds. I think the state is very interested in that. EPA is very interested in that. We just need to get more funding for it. But the only interesting thing I'm going to tell you is that we found one very interesting thing. Cattle egrets feed primarily on grasshoppers. We found the grasshoppers a high content of stigmasterol, and they do not metabolize it. And apparently, cattle egrets do not metabolize it well. So they're excreting high concentrations of one stigmasterol, one fecal sterol. So that could be our indicator. If we go to waters and sample and look for that and use stabilized technique to predict the potential. We had to do some modeling and use the brains of Dr. Grant and others to come up with a prediction at the big level. But this is something that we want to explore in the future. All right, so teaching. I've been teaching 3 or 4 these last 6 years. And then in the fall, several grounding courses, 639, 627, 628. That's basically what I've been doing like 6 years. In terms of service, I think coming from the federal government, I was used to do a lot of service, so I think I did not learn not to say no, and that's okay. I've been in the faculty senate for 6 years. This year I said no, I'm not going to continue. I was in 2 committees that I think very important. I've encouraged any of you to participate. Is the what is it? Committee on Academic Freedom, Responsibility, and Tenure. I've been in one case, very interesting case. The other is let's see, Academic Civil Rights Investigation Committee. I participated in those 2 interesting cases. So those are really committees that occasionally meet, but you have to deal with some very interesting issues, and I learned a lot from those. Also, of course, I was on the undergraduate curriculum review committee, and I don't have to tell you the story what happened there, but anyway, so we're currently waiting to see what's going to happen. Outside the university, I've been an associate editor for Ecotoxicology for years. I participated in several NSF panels. I was on the board of directors of SAGNAS, and so on. One of the things that I've been doing now in collaboration with Dr. Fujiwara is being a co-advisor of the SAGNAS student chapter. I've been with the SAGNAS student chapter since we formed it a while ago, probably 10 years ago. But now we're making an effort of taking students to the annual conference all the time. And I think they enjoy it. They have a lot of good time. So we're always looking for money and sources to take students to this conference because they see other people, they participate with others, and have a lot of fun. Last year, I was happy to be recognized by the Society of Environmental Toxicology and Chemistry as a fellow. So that's really nice for me. And then in 2013, thanks to the efforts of Dr. Fujiwara and the SAGNAS student chapter, I received a Distinguished Scientist National Award from SAGNAS. So those are things that I get. Everything I've done here on campus, I couldn't do it without collaboration of a lot of partners. Geosciences, Jose Sericano, Terry Way, Bob Taylor in med school, Tommy McDonald, School of Public Health, Tom Boughton, and Ethan Grossman for stable isotopes here on campus. And then more lately with soil and crop sciences, doing some microbiology and some analytical techniques as well. So I think it's about the right time. So that's what I have. Thank you.
Questions?
Yes,
That's an interesting method where you did the clustering of the isotopic data and then ran the mixing.
Yeah.
Have you ever seen that before in the literature?
No, and I discussed that with Tom Boughton because you know, I wanted to make sure that I'm just not missing everything. He thought that was a very interesting approach, and he asked me the same thing. He said, no, I haven't seen that. I just thought it made sense because the data are all clumped.
That's always the problem, right?
Exactly. Exactly. But the nice thing when you analyze this statistically, it separates the values nicely, and then that would be a lot easier for the model to tell they're eating from this cluster.
Well, it helps run the mixing model. So what I'm wondering is, I wonder what would happen if you had just taken the average of each of your prey categories, rather than do the clumping based on isotopic ratios. What if you just average for spiders, insects, C3 and C4 plants?
You still
mixing model that way.
you have too many sources still.
Well, you have all this uncertainty, but I just wonder what that result, how that would compare to the estimates you came up with. Because after you do the cluster and the mixing model, you got to go back
Exactly.
and reinterpret.
And I could tell you why.
Cool to compare this.
And I could tell you why I thought this was a better approach, because for a while Dr. Silva was saying, oh, they eat a lot of rice, they eat a lot of rice, and then at the refuge saying, well, there's no rice around here, they hardly go to the rice. So I had to analyze rice, and then I put rice in the model separately. Guess what? The model tells you they're feeding on rice, 100%. So the problem with this model is if you tell them this is a source, it's going to go and look for that specific, okay? But if you tell them all these carbon values and nitrogen values are a potential source, then it will tell you based on the values.
So one thing that's always good to do is if you can eliminate a source based on other knowledge, other evidence, toss it out, and then sometimes that helps the resolution of
Yeah.
the remaining one.
See, and that is true. Here we had a massive problem in the sense that it is surprising that the refuge, they really don't have a categorize the potential food sources. We look at the literature and the values or the plants that Lemon says that they're feeding on. I took the stabilized values of those, and they're way, 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 way down. If I put those as the only source, the model will not even work, because it's basically eliminating all the insects and all the other values. And to me also the other positive thing is the analysis of that fecal material that was collected in winter that is showing all those insects in there. I mean, that cannot be wrong. So they are finding them, they're feeding them. But yeah, that's I presented this model to Kit Hobson, and he and we were discussing some ideas, saying, yeah, you have lots of data there. I had to figure out.
Very interesting.
Yeah. There's another question over there. Just for
Where is that? Cruising stuff?
yes,
Yeah.
right away.
So you showed one slide, and I forget the class of chemical, but you showed a whole bunch of them, and you showed what they're used for,
Hmm.
like fabrics and, I don't know, furniture.
Yeah.
Packaging.
Yes.
But then you said they're toxic,
Yeah.
and yet some are apparently still in use.
Oh yeah.
So why do we do this?
Well, what happens is presumably they, in plastic, for example, they are attached to the structure. But if they were going to dissolve or eventually get released, that's when they will become toxic.
We really want these things.
You may remember the problem of the baby bottles that were used that they put them in the to clean at high heat, and they were releasing some of those bisphenol A and all those things. So that's what happens.
So some have been banned, but some haven't. And you
Oh,
say
yeah.
they're toxic.
That's a long yes.
This is normal?
That is never never going to disappear.
Did you say the EU had banned some of these though?
All the neonicotinoids, except for nurseries, because of the problems with pollinators. Yeah, they placed a moratorium on them for 2 years, but last year they say, let's say for agriculture. Actually, some countries are challenging that because that was a EU decision. But there was a big report of the what is that, Bird Conservancy in Washington. They really wanted to take action because they think that they are responsible for declining grassland populations in the U.S. But the thing is, Gary, is we always talk about all this habitat loss and whatever, but when it comes to contaminants, politically it's a serious issue, I guess. I don't know. Yes, Perry?
So if you stop using them, what's the time to sort of like extinction to the background? How long do you expect this before you actually come down to acceptable levels if you have to start complying?
Are you talking about the neonicotinoids?
Yeah.
I think it will be fast because they're not really that long-lived.
Okay.
They, 5, 10 years, you probably won't see any residues. But of course, the aquatic environment, there are papers that say they're decimating aquatic invertebrates because they're having a lot of issues with that.
Okay.
So,
why are we still seeing DDT effects now? As the same issue?
Very persistent. I think our chemists underestimated their persistence in the environment. Still out there.
DDE.
DDE and isomer or DDT. Oh, and also we have long stockpiles somewhere in your house or whatever, you know. It's